The title page of a Microsoft Teams meeting, ODS seminar series, Bringing Botanicals to Clinical Trial.
Good morning. Welcome, everyone. We'll get started in a minute or two.
A title slide, Office of Dietary Supplements Seminar Series. ODS.od.nih.gov. A logo for the National Institutes of Health. On the right side of the screen, two presenters' video feeds appear, Amala Soumyanath and Barbara Sorkin, NIH. These video feeds sit above a collection of icons representing individual audience members' initials.
Welcome, everyone. I'm seeing the number of attendees is not increasing rapidly at the moment, so I'm going to go ahead and start with my introduction. I am Barbara Sorkin. I am in the Office of Dietary Supplements here at the NIH, where I co-direct the consortium advancing research on botanicals and other natural products. And I'm delighted to be the organizer of the Office's seminar series. Before I introduce today's speaker, a few brief announcements. First, the coming attractions. Our next ODS webinar will be on Wednesday, May 21st at 11 a.m. U.S. Eastern Time. It will be given by Dr. Preston Manuel, who's one of the investigators in that carbon program that I just spoke about. He actually is in the High Content Functional Annotation of Natural Products project of that consortium, and he will be talking about advancing botanical dietary supplement research through collaboration, specifically the mass spectrometry metabolomics collaboration. If you're interested in nutritional metabolomics, it's not as dry as it sounds. I think you'll be really interested in some of what you can learn from his presentation. And if you're not on our seminar's mailing list and you'd like to be, please see the chat. We will be adding an email address where you can request to be added to that mailing. It's also going to be on my next slide.
Text. ODS Event Guidelines. https://ods.od.nih.gov/about/ods_event_guidelines.aspx. Attendees are expected to uphold the professional and educational purposes of meetings by respecting the rights, views, privacy, safety, and dignity of all people in their speech, postings, and other actions. Keep all comments relevant to the event topic. Refrain from engaging in harassing speech or other inappropriate behavior. Posting insulting, obscene, discriminatory, or threatening comments. Disrupting the conversation, including with spam or repetitive posts. Use of AL-based or AL-assisted note-takers. If you see or experience a violation of these guidelines, please contact ODS at ods@nih.gov or send a private message to the meeting host. Participants who engage in any of these behaviors will be given a warning from event organizers, asked to stop the behavior and be expected to stop immediately. Participants who continue to display inappropriate behavior after being given such a warning will be removed from the event.
And this describes the etiquette and respect that we expect from everyone who is participating in our seminars. So in the interest of audio quality, you've all been muted on entry. And we ask that you stay muted until the final questions and answers after the presentation. You do have access to the chat, and we encourage you to put any questions you have for the speaker in the chat as they arise. If I see any that seem urgent, I'll try to get her attention and ask them during the talk. Otherwise, we will take as many as we can fit at the end. Please also use the chat if you're having technical problems with the webinar audio or visual, or the closed captions, and one of us will try to help. And then the crucial caveat, are we advancing? Let me, there we go.
Text. Any opinions expressed by ODS seminar speakers or attendees do not necessarily reflect the views of the National Institutes of Health, the Department of Health and Human Services, or the United States government. ods.od.nih.gov.
So, with that, it is my pleasure and honor to introduce today's speaker, Dr. Amala Soumyanath. Dr. Soumyanath is a professor in the Department of Neurology at Oregon Health and Science University and a pharmacognosist. That is someone who studies the health effects of plants. Amala received her pharmacy degree and Ph.D. from the University of London. Her current research focuses both on understanding the biological effects of chemically complex plant-derived products and on the development of plant-derived drugs. For some time, her research has focused on the potential of botanical-derived products to promote resilience to neurological decline in aging. I'm pleased to say that some of her funding has come from an award partly supported by the NIH Carbon Program. And as I'm sure she'll tell you, that program is a collaboration at the NIH and between the Office of Dietary Supplements, the National Center for Complementary and Integrative Health, and the National Institute on Aging. So with that, Dr. Soumyanath, the microphone is yours. I will, let's see, stop sharing. There's the button.
The two presenters' video feeds fill the screen.
And look forward to your presentation.
Okay, well, thank you very much for that wonderful introduction. It's really an honor for me to present to this in the seminar series because we've done a lot of work on botanicals, and I have to say the vast majority of that has been funded by the NIH. So being able to present at this forum is truly an honor. So with that, let me start sharing my slides, and hopefully that is working. And I'll go into presentation mode. Let me change the display settings. Is that good?
Perfect.
A title slide. Bringing Botanicals to Clinical Trial, Groundwork and Challenges. Amala Soumyanath, Pharm, Ph.D. Professor, Department of Neurology. Program Director, Benfra Botanical Dietary Supplements Research Center. Oregon Health and Science University. Portland, Oregon. USA ODS. Seminar, April 23, 2025.
Okay, wonderful. So let me get my pointer going, and then we can get started.
A title
so
slide.
As you heard, I'm a professor at Oregon Health and Science University, talking from sunny Portland, Oregon today. It's a beautiful day out there, and I'm very pleased to be able to share our work with you here.
Slide. An image appears of a scientist in a laboratory wearing gloves, surrounded by plants. Text, Benfra BDSRC, quote, Botanicals Enhancing Neurological and Functional Resilience in Aging, an NIH-funded Botanical Dietary Supplements Research Center.
I'm the program director for a part of the carbon program that you just heard about, and we've acronymed our center the Benfra Center. So Benfra is an acronym for Botanicals Enhancing Neurological and Functional Resilience in Aging. And as you just heard, that's been the focus of my research, and particularly this NCCIH and ODS funded botanical center that we have here at OHSU.
Slide. 4 logos appear beside images depicting the organizations, a mix of academic buildings and a field worker.
It is in fact a collaboration between 4 different organizations, not just OHSU, but also Oregon State University, a company called Oregon's Wild Harvest, and the University of Mississippi. But as I said, I'm based at OHSU, and I'll talk about some of my collaborators as we go through the talk.
Slide. Aging and cognitive decline. A bulleted list of text compares normal aging and examples of pathological aging, including Alzheimer's disease and Lewy body dementia, Parkinson's, vascular dementia, and other. A line graph depicts mean T-scores versus age for different age-related conditions over a lifespan of years from 20 to 90, including inductive reasoning, spatial orientation, numeric and verbal ability. All show a decline around age 60.
So what we're looking at is whether botanicals can help functional resilience in aging or be used to develop drugs to treat the kind of neurological decline that one might see during the process of aging. And I think we're all familiar with the concept that even in normal aging, we often face aspects of cognitive decline, and there's different aspects to this, all of which are known to decline at different rates, even with normal aging. And of course, if you're very lucky, you may not have any kind of decline whatsoever. As well as normal aging, there is, of course, pathological aging. And in this case, one of the most common causes of cognitive decline is Alzheimer's disease, although others also exist, as I've listed here.
Slide, Botanicals Understudy at Benfra. Images depict two different green plants as well as a supplement bottle. The images are captioned Centella asiatica, urban, Goticola, and Withania somnifera, Dinyl, ashwagandha. Text: both herbs are highly regarded as rasayana or rejuvenating herbs in Ayurveda with reported benefits on memory and nerve health.
Now at the Benfra Center, we're focusing our research on two specific botanicals, Centella asiatica, which is sold as the dietary supplement Goticola, and Withania somnifera, which is sold as the dietary supplement ashwagandha. Now, why did we choose these herbs? It's because both of these herbs are highly regarded as rasayana herbs or rejuvenating herbs in the Ayurvedic tradition. And they also have a lot of both scientific literature and reputation, if you like, for reported benefits on memory and nerve health. So this was the starting point for our work.
Slide. G 1994, Dietary Supplement Health and Education Act. A list of bulleted text items describes the act.
And I wanted to just give you a little bit of regulatory background to dietary supplements before I start talking about the research that we've done. As most of you are probably aware, the dietary supplements that we can buy in stores and so on are governed by the or regulated by the Dietary Supplement Health and Education Act of 1994, otherwise known as DSHEA. And under these regulations, dietary supplements are neither foods nor medicines, and you don't really need to have any kind of proof of efficacy in order to market them. And because of this, there are also no standardized preparations. And they don't need to be FDA registered, although the FDA does need to be aware that that you're marketing a product. And one of the important factors is that you cannot make any therapeutic claims about your product on the label. And in fact, you have to have a disclaimer on the label that says that these statements have not been evaluated by the FDA and the product is not intended to diagnose, treat, cure, or prevent any disease. So while you can't promote them for any therapeutic functions, equally you have to also make it clear to the public that they haven't been evaluated.
Slide. Several images depict Centella asiatica, urban, Goticola dietary supplement examples. Text: roughly 1,500 registered DS product labels. https://dsld.od.nih.gov/dsld/index.isp.
So because of this, for any given botanical, you tend to have a very wide range of products. So this is just some examples of the different types of products that you can have from Centella asiatica, for instance. I'm just going to try and make sure I can see people here, but I hope that doesn't affect there we go. Okay, so you have a wide range of different products here from a given botanical. And so as you can imagine, these are not all going to be the same. And I'll come on to that point in a second.
Slide. Do botanical dietary supplements work? Bulleted text provides descriptions for preclinical research, clinical research, and problems in evaluating evidence.
Now, a lot of people would really like to know, do botanical supplements actually work? And the thing is, it's kind of difficult to give a straight answer to that. They may work, and as an individual, when you take a botanical product, you may feel that it works for you, which is great. But when you ask it as a very general question, like, does Centella asiatica work for, to treat neurological decline or a cognitive decline, the answer kind of has to be, it depends. It depends on the product. And what you find if you look in the literature is that there tends to be a lot of preclinical research on a lot of botanicals, including in vitro and in vivo studies. But clinical research, in other words, clinical trials, which have really proved efficacy or shown assessed efficacy, are limited or non-existent for many herbs, for the majority of herbs, in fact. And even the studies that have been done often lack scientific rigor. And another problem is that although there might be a lot of studies, you find that it's very hard to come up with a definitive answer based on the bulk of the evidence, because very often studies are done with very poor description of the products used in terms of their chemistry or even their origin sometimes. And also, because of that, you find that products used from the same herb used in different studies show a lot of variability. So different products are used in different studies. And what we need to remember is that whenever you're looking at a study on a particular product, the data generated is really specific to that product. And so another, if you had a different product from the same herb, like I have here, like if you did your study with this product, the results may not apply to that product there because they will have chemical differences or compositional differences.
Slide, Conventional Medicines versus Botanical Products. A bulleted list of text items contrasts the differences between conventional and botanical.
And the other thing that you need to know about botanical products, which makes, again, some level of complexity in understanding the scientific data, is that they are very different from conventional medicines, which tend to be single chemical entities, whereas botanical products are a mixture of a vast mass of chemicals or individual chemical substances, and that the activity of that botanical is likely due to more than one active compounds and potentially even due to interactions between them. Another thing that makes botanical research both challenging and exciting is that the chemistry of a given product can show variability due to genetic, geographical, cultivation factors, and as well as processing methods that have been used to make that specific product. And it's a fact that we probably, for the vast majority of botanicals, don't really know the activity of all the constituents and exactly what is contributing to activity. And so when we do these studies, we have to kind of bear this in mind and know that you're dealing with a very complex product. And I'll hopefully give you some examples of how we've dealt with that.
Slide, Recent Publications on Optimizing Clinical Trials Evaluating Natural Products. Two titles appear. First, Improving Natural Product Research Translation, From Source to Clinical Trial, written by Sorkin et al., The FASEB Journal 2020. Second, National Center for Complementary and Integrative Health Perspectives on Clinical Research Involving Natural Products, Weber, and Hopp, Drug Metabolism and Disposition, 2020.
Now, it's very important, of course, to show efficacy, to do a proper clinical trial, efficacy trial, but it's not so straightforward. And the NCCIH and ODS have really been looking at what is the best way to do clinical trials, taking account of all these complexities that I've just described.
Slide, Roadmap for Natural Product Clinical Trials proposed by the NIH National Center for Complementary and Integrative Health. An image depicts examples of building blocks of natural product clinical trials, with stacked blocks full of text. The foundation is historical use and fundamental preclinical knowledge, and the blocks above include text like pharmacokinetics, efficient recruitment and retention, and then the top block reads multi-site efficacy trials.
So they've come up with a roadmap for doing clinical trials, so that it's not just a case of you pick a product and you go straight into an efficacy trial, which can often be very expensive, but that you do the work very systematically so that you have a good chance that your answer that you get when you do your efficacy trial is a valid answer. So some of the building blocks to this is it's all based on a foundation of historical use and or fundamental preclinical knowledge. So try to base your study on some kind of background which suggests that you have a likelihood of efficacy in your efficacy trial. And then it's important to pay attention to things like product quality, its composition, and select the right kind of outcome measures and population and so on to do your study. And I'll go into some of these in a little bit more detail in the course of the talk. But the overall point is to say, don't just rush straight into an efficacy trial. Try and build up an evidence base so that you can design a good trial based on your earlier studies, and which therefore will give you a good chance of really evaluating whether the product you're testing is efficacious or not.
Slide, BENFR pipeline for optimized clinical trials. A narrow links text, from product authentication and optimization, to identification of active compounds and mechanisms, to evaluation of in vivo efficacy and translationally relevant biomarkers, to optimally designed clinical trials. The entire arrow points to valid evaluation of botanical efficacy.
So at the BENFR Center, we conceptualized this pipeline of research for optimized clinical trials. And this it begins right from the beginning, where you have to make sure that you have methods of making sure the product you're testing actually really is the herb that you from the botanical that you think it is. So product authentication and optimization. It's very important that we know what the active compounds are and have a good understanding of their mechanisms, because if you know what the mechanisms are, you can go look at biomarkers. So also, of course, before you go into human studies, evaluate the efficacy of your botanical using in vivo methods, and then look for signals or mechanisms within your in vivo model that you can then look at in a human. And this is what we mean by translationally relevant biomarkers. So before you do your clinical trial, make sure that whatever mechanisms you saw in your in vivo system, in your animal model, can actually be seen in a human, and then you have a better chance of hopefully detecting an effect in your clinical trial.
Slide, research pipeline for rational phytotherapy. On the image of the same arrow, the text ashwagandha appears, pointing to an early spot on the arrow. The text Botanicals appears, pointing to a spot toward the tip of the arrow.
So when we started the BENFR project in 2020, we had already done a fair amount of work with Centella asiatica or gotu-kola, and whereas ashwagandha was a new herb for us. So for the majority of my talk, I'm really going to be focusing on the work that we've done with Centella asiatica, and hopefully there'll be time at the end to tell you a little bit about where we are with ashwagandha as well.
Slide, preclinical studies on Centella asiatica for Alzheimer's disease, ASHU/OSU. A flowchart shows the source material, water extraction process, and various research paths including active compounds analysis, in vivo studies with mice and Drosophila, in vitro cell studies, and safety evaluations. Chemical structures and analytical data are included for key compounds.v.
So this slide summarizes sort of over almost 15 years of work that we've done on Centella asiatica. And I'm not going to go into this in a lot of detail, except to say that when we began our work on Centella, we were really looking at it as a possible treatment for Alzheimer's disease. And we began our work by making a water extract of the herb, which I'm going to call CAW throughout this presentation. And the reason we chose this extraction method was based on prior scientific evidence that suggested that that was the best way to bring out the active compounds of this plant. Now you might say, wait a minute, you just told me that dietary supplements cannot have therapeutic claims, and now you're mentioning Alzheimer's disease. Well, the thing is that the FDA does allow us to develop botanical drugs. So you can, aside from a dietary supplement, there's another classification which is known as a botanical drug, and in this case you can make a therapeutic claim if you show efficacy, and the approval process to have something designated as a botanical drug is very similar to what we have for conventional drugs. So as I said, a lot of our work has centered on looking at it for looking at Centella asiatica as a potential treatment for Alzheimer's disease, but as you'll see, it has overlaps with its use as a dietary supplement, which is more to just provide resilience against cognitive decline in aging.
Slide, pathology and clinical features of AD. A diagram illustrates the progression of Alzheimer's disease. It shows how neurofibrillary tangles and beta-amyloid plaques lead to loss of synapses and neurons. This causes cognitive and behavioral symptoms, including attention deficit, memory loss, depression, and hallucinations. A brain comparison depicts a healthy brain versus one affected by Alzheimer's. Text, http://www.galandos.com/index.php/en/science-and-technology/ad-pathology.php.
So just very briefly about Alzheimer's disease. I know that most of you are probably familiar with this because this is a disease that receives a lot of publicity and there's a lot of anxiety around it as well, of course. But one of the main features of Alzheimer's disease is the presence of these beta amyloid plaques. Beta amyloid is a protein. There's been a lot of controversy over the years as to how causative this is as a primary cause of Alzheimer's disease, but it remains a fact that anybody who has Alzheimer's disease has these amyloid plaques, abnormal plaques of amyloid protein in their brain. And they also have another protein called tau, which is hyperphosphorylated in an abnormal way and is found within neurons as tangles. There's also deficiencies in neurotransmission because of receptor deficiencies and so on. And these are some of the causes that lead to basically loss of synapses, which are the connections between neurons, and loss of neurons themselves, and also a lot of the cognitive and behavioral symptoms that we see. So when we started looking at the possible effects or possible benefits of Centella asiatica in Alzheimer's disease, that's where we began.
Slide, collaborators, preclinical studies on Coitashu. 8 photos appear of Nora Gray, Ph.D., Joseph Quinn, M.D., Kirsten Wright, North Dakota, Doris Kretschmer, Ph.D., Maya Caruso, B.S., Don Matthews, Ph.D., Jonathan Zweig, B.S., Christopher Harris, B.S.
And I want to begin by saying that this has been a very long process with a lot of folks, and I'm really grateful to all my collaborators. It's definitely required a lot of multidisciplinary expertise, and everybody whose pictures you see has contributed in some way. But I would like to specially mention Dr. Joe Quinn, who's a neurologist at OHSU, who's been supportive of this project throughout, and he has expertise, right from bench to bedside. And Dr. Nora Gray, who's done a lot of the animal in vivo work, Dr. Kirsten Wright, who's done a lot of the helped with a lot of the translational studies, and Dr. Doris Kretschmer, who's our expert in Drosophila studies, and of course all the wonderful research assistants that we've had as well.
Slide, CAL protects neuroblastoma cells against beta amyloid cytotoxicity. 2 bar graphs labeled MC65 and SY5Y are displayed side by side. Each graph shows 4 columns representing different experimental conditions. The y-axis measures % survival up to 150% viability. Below the graphs, text explains the experimental setup involving CAR treatment. Note, in MC65 cells intracellular beta amyloid is induced by removing tetracycline from the culture medium.
So going back to our very first experiments, we're using a very simple cell culture model. So you can grow neuroblastoma cells, and these are just simple cell lines. And under certain circumstances, you can actually expose them to beta amyloid, these plaques, the protein that forms these plaques that I mentioned. And what you find, so that you'll see a bunch of these graphs coming up, where essentially the first bar is the control, where we just grow the cells normally. The second bar in each case is just the cells growing in the presence of our CAW Centella extract. The third bar is where you have beta amyloid present, and the fourth bar is where you have both beta amyloid and the Centella extract present. And what we're measuring in this experiment is viability, and I hope you'll see that as soon as you have beta amyloid present, viability of these cells goes down. But if you have Centella present as well, that somehow protects these cells from the toxic effects of beta amyloid. So this was very exciting.
Slide, CA activates genes associated with the antioxidant response element. 2 side-by-side bar graphs are labeled MC65 and SY5Y. Each graph shows multiple sets of bars representing different conditions at 24-hour and 48-hour time points. The bars vary in height, indicating different levels of fold change across the conditions and time points. Error bars are visible on top of each bar. A note, NF2L2, also known as NRF2, GCLC, HMOX1, and NQ01 are genes involved in the antioxidant response element.
And we also wanted to try and understand what the mechanisms were. And again, there's a lot of very complicated looking graphs that I'll just try and give you the take-home message from because of lack of time to go in a lot of detail. But what we did find is that in these cells, when you compare the different treatment conditions, one of the things we saw is that Centella asiatica was able to activate the genes that were associated with antioxidant response. and, in other words, give the cells an ability to respond to oxidative stress.
Slide. CA induces the expression of mitochondrial ETC genes. Two bar graphs compare data for MC65 and SY5Y. Each graph has 4 groups of bars representing different conditions. The y-axis shows fold induction, while the x-axis lists various time points.
We also found that Centella asiatica induces the expression of mitochondrial electron transport genes. So what are these electron transport chain genes? The electron transport chain within mitochondria is the mechanism by which ATP is produced and produces energy for the cells. And we did find that beta amyloid, the third bar along, would decrease the functioning of this chain, and this was able to be reversed by the presence of our Centella extract. So these were 2 mechanisms which seem to be relevant for the protective effects of this extract.
Slide. What are the active compounds in CA?
And so our next thing was what are the active compounds that are caused in the extract, which is a crude mishmash of substances. What are the active compounds within this extract which are causing this effect?
Slide. Triterpenes, not neuroprotective in MC65 cells. A chemical structure of a triterpene compound is displayed alongside tables and graphs. The tables show compound compositions and CMC values. 2 bar graphs illustrate the effects of different concentrations of AA and S on cell viability. A caption explains these triterpenes are the best known compounds in CA, with AA and S showing neurotropic and neuroprotective properties.
And the first type of compound that we looked at were the triterpenes. Now, if you look at any literature on Centella asiatica, it will tell you that triterpenes are, in many cases, the active compounds, which you might expect, because they're very unique. They're somewhat unique to Centella asiatica, and they're also quite abundant. So we tested the effect of these different compounds on viability of these cells, and we were surprised to find that the triterpenes, in fact, were not the components that were responsible for this protective effect. And that was kind of serendipitous because it made us look further.
Slide. CA contains caffeoylquinic acids, identified in conjunction with OSUMS facility. An image contains a table listing various compounds and their CA composition percentages. Below the table are chemical structures of monocaffeoylquinic acids and dicaffeoylquinic acids. The structures show molecular diagrams with carbon rings and side chains labeled for different acid compounds.
And we also found that the extract contains these compounds called caffeoylquinic acids, which I'm going to abbreviate as CQAs from now on. And we found that the extract also contained these compounds.
Slide. CQAs are neuroprotective in MC65 cells. A bar graph displays the dose-dependent protection for various concentrations. Multiple colored bars represent different compounds or conditions. The y-axis shows the percentage of protection, while the x-axis lists different treatment groups.
And when we tested those for their neuroprotective effects, lo and behold, the CQAs were the ones in the extract that were responsible for this protective effect. So the lesson here for us was not to always go with the obvious constituents, but to always look further and see if there might be something else in there which is actually adding to or actually causing the effect.
Slide. CA also improves mitochondrial function and decreased oxidative stress in mouse primary hippocampal neurons exposed to beta-amyloid. A research article appears, Centella asiatica attenuates mitochondrial dysfunction and oxidative stress in AB-exposed hippocampal neurons.
So then we moved on to more complex and higher models, if you like. And so the next set of tests we did was with these primary hippocampal neurons. So these are freshly taken out of a mouse. And so that's a lot closer, if you like, to the in vivo situation. And we found that the same effects on mitochondrial dysfunction and oxidative stress were observed in this model. I'm not going to show you any graphs, so please take it from me and look at this publication if you're interested in this.
Slide. CA, DTs, and CQAs stimulate dendritic arborization in mouse primary hippocampal neurons. 2 brain cell illustrations show different conditions. Bar graphs present quantitative results from experiments. Labels indicate various treatments and measurements.
And the other very interesting effect we saw in these hippocampal neurons was that when you add CAW, you get this increase in branching or dendritic arborization. And this property has typically been associated with improved cognition, and also it would be something that would be expected to increase the synaptic connections between nerves. So we found you can quantify the dendritic arborization by a process known as shoal analysis, and we found that not only did the extract, but also the caffeoalquinic acids and the triterpenes also had this effect on increasing the dendritic arborization. And this was true of the mixtures as well as individual CQAs and triterpenes. Not all the CQAs had an effect, but in fact, the 4 triterpenes that we had all had this effect. So again, whether something works or not probably also depends on your endpoint of what you're looking at. What are you actually measuring? This was an interesting effect to see that if we combined the triterpenes and the CQAs, that actually seemed to decrease dendritic arborization, which was a bit surprising, especially given that this mixture of triterpenes and CQAs is present in the entire CAW. But what that told us is that while this mixture in this particular assay might be inhibitory, there are other things in the centella extract that are also contributing and making the whole extract actually biologically active. That's all very complex, and I'm not sure we still understand exactly what's going on, but that's all the subject of additional work that we've been doing at the Benfra Center to try and unravel.
Slide. Targeted and untargeted analysis of phytochemicals in Centella asiatica extracts by liquid chromatography and high-resolution mass spectrometry, OSU Mass Spectrometry Center. 6 photos appear: Claudia Meyer, Ph.D., Fred Stevens, Ph.D., Armando Alcazar, Magana Ph.D., Liping Yang, Ph.D., Jay Wu Choi, Ph.D., Luke Marney, Ph.D.
Okay, now all this is possible because we have excellent analytical support and from our collaborators at the Oregon State University Mass Spectrometry Center, led by Dr. Claudia Meyer and Dr. Fred Stevens, and they're able to do all the analyses and so on that we need in order to analyze these plant extracts.
Slide. Targeted and untargeted analysis of CAW compounds. 2 graphs appear. On the left, targeted detection of individual CQAs and TTs of interest by LC multiple reaction monitoring mass spectrometry. On the right, principal component analysis of 5,512 m/Z features from LC high-resolution tandem mass spectrometry, LC-HRMS/MS, of CAW made from 8 accessions of Centella asiatica, CA1-CA8. Both are credited, Alcazar-Magano et al., Phytochemical Analysis, 2020, Yang et al., J. Crom. Open, 2023.
And they have 2 types of analyses that they do, both targeted and untargeted analysis. By targeted analysis, what I mean is and all of these use liquid chromatography coupled to mass spectrometry techniques. So in targeted analysis, you are actually setting your parameters, your analytical parameters, to detect specific compounds, whereas in untargeted analysis, you can, if you like, get a fingerprint of everything that extract contains. And that's important for things like botanicals, where, as I've just said, the activity may not be because of a single chemical or known chemicals. It's nice to be able to get a fingerprint of everything that is in a particular extract. And using this untargeted analysis, you can do things like principal component analysis, where you basically are able to compare different extracts made from different products, different batches of Centella asiatica and so on. So that's what was done here. So this was, you can use this for principal component analysis to compare CAW made from 8 different sources of Centella, for example.
Slide. Relevance to human use. An image of a beaker points to the left and right. On the left, an image of petri dishes, labeled, biological effects seen when added directly to cells at known. On the right, an image of an older man, labeled, active when taken as an oral supplement/drug. Digestive fluids/enzymes, gut microbial metabolism, absorbed into bloodstream. Liver metabolism, reach target tissues. Concentrations. Sex differences.
So we've done these in vitro studies, and of course now what's really important is how is it relevant to human use? And we need to be aware that, obviously adding something directly to cells, and I'm sure I'm stating the obvious here, is not the same as giving it to a human, because when someone takes a dietary supplement orally, whatever is ingested is subjected to everything that goes on in the gut, and including microbial metabolism and so on. If it gets absorbed into the bloodstream, it may or may not be. Then we also have to look at liver metabolism. Do the compounds reach target tissues? What concentrations? Are there sex differences, and so on?
An image of an insect and mouse appears. Labeled in vitro models can provide some answers.
And so we can't go directly from cells to humans, and what often helps to give us some answers are some of the in vivo models that we can test in that might provide some of these answers.
Slide, assessing locomotion and cognition in Drosophila, fast phototaxis assays. A set of test tubes is arranged in a rack. Two graphs display performance in a phototaxis assay for males and females across different age groups. The graphs show a declining trend in performance as age increases for both sexes.
So I want to mention that we've done some work with Drosophila flies, and that might seem a little strange, but in fact, Drosophila models can be very useful to show us some effects on humans, and we can discuss maybe later what the benefits and limitations are of Drosophila. But what we found with Drosophila flies is similar to humans. They do show a decline in neurological function with aging. And so there's a simple phototaxis test that you can use, which measures their response to move towards light. And you find that the older they get, they do actually decline in their ability to respond to light.
Slide. Call provides resilience to age-related decline in Drosophila phototaxis. A bar graph displays percentages for 4 groups, with 2 bars showing higher values around 60 to 70% in males and females, as compared to the control groups. An image of 3 test tubes contain layers of liquid and fruit flies. Text below explains an experiment involving Drosophila flies, regular food, and CAW treatment over several weeks.
And whereas if you treat them with CAW extract, then you find that you're able to restore some of this ability to respond to this phototaxic response. So this was very exciting, and it gave us a simple way of, again, testing things in a simple in vivo model. And so these are the flies. We mix the centella extract in their food, and when they were treated with this for 2 weeks, they did show this improved phototaxic response. And again, this is a simple model where you can go in to test different compounds. And what we found, again, was both the triterpenes given alone, the CQAs given alone, and the combination of TTs and CQAs also had this ability to restore phototaxic response in these aging Drosophila flies. So once again, showing that these compounds are important for the biological activity of the centella extract.
Slide. In vivo mouse models of aging and ADTG 2576 mouse develops amyloid beta plaques and cognitive impairment with age test at 20 months. 5s-fed mouse rapidly develops amyloid B plaques and cognitive impairment, test at 7 to 8 months. Aged mice, wild type, test at 18 to 20 months. Test paradigms: Morris water maze, novel object recognition, object location memory, odor discrimination learning reversal, condition fear response.
Then we moved on to mouse models. I say we moved on to this, but a lot of this work has been going on in parallel. And we've tested the centella extract in both models of Alzheimer's disease, which have these beta amyloid plaques, and simply mice that are aging. So these are just aging models, and done a bunch of different tests for cognitive function. And I will just give you a couple of examples of studies that we've done, because there've been a lot of studies, and I'll give you a link to some publications later.
Slide. Mouse study protocol, 5s fat and weight mice. 2 mice are depicted at the top of an image. A timeline shows the progression of an experiment over 5 weeks. The timeline indicates when treatment is initiated, cognitive testing starts, and mice are sacrificed for brain, blood, and tissue collection. The experiment compares a control group to a CAW-treated group.
So our model for doing these studies has basically been to treat mice, either give them their usual drinking water and diet, or put this centella extract in their drinking water at different concentrations to get different doses. And we treat them for a period of about 5 weeks. And we do the cognitive tests starting at 2 weeks. The test can take a couple of weeks to perform. And at the end of this treatment period, you can take tissues from your mice and then analyze them to get, again, an inkling of what mechanisms might be taking place.
Slide. CA produces a dose-dependent effect on CFR in 8-month-old male and female 5's fat and weight, normal, mice. A diagram illustrates a conditioned fear response paradigm. 3 boxes depict experimental stages: exposure to context, onset of shock, and testing. A graph shows contextual freezing percentages for different groups over increasing LP's treatment. Statistical analysis results are presented, indicating significant differences between groups.
So here is a simple test that we do. This is conditioned fear response. So in this case, this was using a mouse model of the 5XFAD as an Alzheimer's mouse model, or just wild-type mice from the same litter mates that were bred during the breeding process. And in this test, what you do is you start off by putting a mouse in a chamber that has a wire mesh. You apply some electric shocks to this mice, which is, of course, an unpleasant experience. And then you put the mouse back in that same chamber after 24 hours. Now, if it remembers that it had a shock the last time it was in there, then it will freeze. And what you can do is measure the amount of time it spends freezing when it goes back into that same context. And so this shows the time spent freezing as a function of the dose of CAW that these mice received. And I think you can see immediately that the more CAW they had, the more time they spent freezing, which indicates better memory. And other things that we noticed were that, of course, the 5XFAD mice in general, so the red is the females and the blue is the males. And we found that the 5XFAD mice in general had poorer memory than the wild types, which is what we'd expect. We saw a definite dose effect, and that females generally showed more freezing than males, and this has been known by others. But we didn't see any gender differences in the effects of CAW. So this was kind of exciting to see that we were able to see this dose-dependent effect of the centella extract on memory in these mice.
Slide: mouse study to examine effects of TTs and CQAs. Arrows on a 5-week timeline indicates the start of cognitive testing in the mice sacrificed for brain, blood and tissue.
And so the next thing was to examine the effect of the triterpenes and CQAs in this model to see if we could see which of these compounds were responsible. Now, because it's kind of expensive to buy these pure compounds and put them in the water, in the drinking water, most of which we then throw away, we decided that we would include everything in the diet of these mice.
Slide: CA, CQA and a TT/CQA mixture in the diet attenuated conditioned fear response deficits in male 5's fed mice. 2 bar graphs, for male and female, are labeled, Figure 1. Conditioned fear memory evaluation of 5's fed mice, 5X, after treatment with Centella asiatica water extract, CAW, or CA compounds. Contextual fear response, CFR, analysis after 4 weeks of treatment being incorporated into the diet. Matthews, et al., 2020, Nutrients, 12, 3488.
And in this study, we really saw very little effect in females at all. And in males, what we found was, as expected, the 5X mice did have poorer memory than the wild types. And we were able to restore memory a little bit similar to wild type with the CAW, the CQAs, the TTs, and the combination. And particularly the caffeoalquinic acids were the ones that reached statistical significance. But in this entire experiment, we were kind of dissatisfied with the responses that we saw. And we weren't sure what had happened there, except remember we gave the test compounds in the diet rather than the drinking water.
Slide: CA in the drinking water but not the diet improved recognition memory in old mice. 2 images depict differences in training day, with a mouse standing between 2 of the same object, and testing day, where one object is the same, and the other is novel. A graph displays retention data for 4 groups: young 0, old 1000 chow, and old 1000 water. Each group is represented by a scatter plot with individual data points and a mean line. The y-axis shows % time with novel object from 0 to 100. Gray et al., Frontiers in Aging, 2024.
And more recently in Benfra studies, we've also seen something similar. So in this experiment, we're looking at mice, aged mice, not Alzheimer's disease mice. And this is a different test called the Novel Object Recognition Test, or NORT. In this test, you show a mice 2 objects, And then after a while, you take it out of the chamber and you replace one of the objects with a novel object. And then you can measure how much time the mice spends looking at each of these objects. And because mice are inherently curious, they should spend more time with the novel object if they remembered what they saw before and they know what's new this time. So what did we find? If you compare young mice and old mice, young mice, as you'd expect, spend more time, more than 50% of the time, with the novel object. Whereas as mice get old, they, it's kind of middling. It's almost at the 50% point because they don't remember that they've seen this object here before. Now, when we added the centella in the chow, in the diet, we didn't see an improvement from the untreated mice. But when we put it in their drinking water, we did see an improvement. And the doses were designed to be about the same, whether they had it in the chow or the water. So the question was, what is actually happening here?
Slide 5's fed mice at higher plasma levels of TTs, CQAs and CQA metabolites when given CA in drinking water than in the diet. 3 bar graphs display the results of plasma concentrations. Spears et al., Frontiers in Neuroscience, 2024.
And this is going back to the 5x FAD, but we were able to analyze the plasma from mice that either had CAW in the drinking water or in their diet. And what we found that if you measure the triterpenes, the CQAs and CQA metabolites, across the board, if you gave it in the water, which is all the orange graphs, you had higher levels of these compounds in the plasma of these mice compared to what they received if the CAW was included in the diet. So again, another challenge is making sure you're giving these compounds in the right form in your test system.
Slide, similar mechanisms of CA were seen in neuronal cultures and mice. A summary of CA mechanisms lists results for different CA effects, such as synapse gene expression, dendritocarburization, and cognition. Text, increases antioxidant response, mitochondrial activity, synapse health. Publications at www.osu.edu/benfra-bdsrc.
So after a lot of work, and I just want to say that we have a ton of publications which are on our Benfra website, and I'm really just summarizing the data here. So for anyone who's interested, please do visit that website. What we found is that a lot of the mechanisms that we saw, which is antioxidant response, mitochondrial activity, increased synaptic health, when we analyzed the brains of mice that had been treated, we found a great deal in parallel between the in vivo studies and the in vitro studies. And this is not always the case, but in our case, we were fortunate that we did see this.
Slide, multiple pathological processes in AD. The top of a flow chart is labeled aging, genetic factors, environmental factors, oxidative stress. 4 branches are compare the effects of amyloid hypothesis, tau hypothesis, oxidative stress hypothesis, and mitochondrial hypothesis. The last 2 branches are circled, and labeled targets of CA, also involved in non-pathological aging. Omar et al., 2017, J. Nutra, Biochem, 47, 1-20.
And it does seem that the oxidative stress and mitochondrial function are the main targets of Centella asiatica rather than targeting amyloids, which is what some of the current drugs do. And the interesting thing is that these targets are not just involved in neuronal loss in Alzheimer's disease, they're also found in non-pathological aging, which suggests that this might be how Centella could function as a useful dietary supplement.
Slide, roadmap for natural product clinical trials. The stacked blocks reappear, with the foundation circled, labeled historical use and function preclinical knowledge. 3 blocks in the next row up are circled, appropriate population selected, outcome measure selection, product quality.
So going back to this roadmap, at this point, we felt we had kind of covered a lot of these boxes because, we got the preclinical studies. We kind of knew that the product had to have triterpenes and CQAs at a bare minimum, and also knew that, this could be effective both in normal aging and in Alzheimer's disease, and that we should look, we knew what kind of outcomes to look for.
The title of an article appears, Developing a Rational, Optimized Product of Centella asiatica for Examination in Clinical Trials, Real-World Challenges. Wright et al., Frontiers in Nutrition, Volume 8, Article 799137, 2022.
So then we were ready to start our translational studies and clinical trials, and of course this brought on a whole new set of challenges, and we've published some of our trials and tribulations in this paper here.
Slide, plant material selection. An image displays a chart comparing different parts and forms of an herb across geographic locations. It shows bar graphs for malicoside, asiaticoside, madecassic acid, and asiatic acid levels. The chart includes categories for aerial top, leaves and stem, leaves only, and whole herb, as well as different processing forms like cut and sift, decut, and powder.
But this centered around right from the start, selecting plant material. I already mentioned that we had these 8 different sources of centella, which in fact showed vastly different from, chemical composition. So just measuring the active compounds, triterpenes, each of these bars represents a different batch of centella. We needed to get a very large batch to make our clinical trial product. And of course, we wanted to try and match it as closely as possible to the centella extract that we'd used in our mouse studies. And so just as one example, if you look at asiaticoside in the green plant material as opposed to the purple, you'll see how different that is. And what we ended up doing was to actually combine CA3 and CA6 to try and get an average, a sort of middling value for this.
An additional bar graph flashes by.
And it was a similar story with the caffeoylquinic acids again.
Slide, targeted and untargeted analysis of Ca compounds. An image contains 2 scientific graphs. On the left is a chromatogram with multiple colored peaks representing different compounds. On the right is a scatter plot showing principal component analysis results for various samples labeled CA1 through CA8. Both graphs relate to mass spectrometry analysis of plant compounds from Centella asiatica. Alcazar Magania et al., Phytochemical Analysis 2020, Yang et al., J. Crom. Open, 2023.
And then, as I've already mentioned, these, if you did a untargeted principal component analysis, a lot of these different centellas did show a lot of variability in their overall composition. So you can see that these 4 plant materials were somewhat similar, whereas these showed differences. So this is what you're up against, and I think the best you can do is at least try and standardize it to the known active compounds.
Slide, heavy metal and pesticide residues in CA samples. A table displays measurements of heavy metals and pesticides in various samples. The columns show source, cadmium, lead, arsenic, number of pesticide residues, and highest pesticide residue. Multiple rows present data for different raw materials and extracts. Some values are highlighted in red, indicating they exceed allowed exposure limits.
Another issue was in fact impurities, and we found many of the plant materials that we bought and analyzed to see if they could be used to make our products were in fact had levels of pesticides and heavy metals even, and some of these fell outside the acceptable range. So these are some of the challenges that we had to overcome.
Slide, human dose selection. Text, identify a dose that is biological effect in mouse model, Ca 200 to 1000 mg per kilogram per day. Calculate human dose equivalent. Allometric, interspecies, scaling, 2 grams to 10 grams per day. Adjust based upon highest previous human dose of Centella asiatica triterpenes. 2 grams and 4 grams Ca per day. Narab and Jacob S. A simple practice guide for dose conversion between animals and human. J. Basic Clin. Farm. 2016, 7-2, 27-31, Liu, et al. Dermatological Experiences, 2018. 20, Sup 1, No 1, p. 12-22. Online Allometric Scaling Calculator, http://climber.altervista.org/minor/allometry.html.
The next question was, what dose do we give a human? We knew how much you need to give a mouse or a Drosophila fly in order to get an effect, but how does this translate to humans? Fortunately, there is a process known as allometric scaling that you can use to translate the dose, and it looked like the doses we'd given to 5xFAD mice would translate to 2 to 10 grams in a human. We decided to not go above 4 grams because that was similar to the highest dose of triterpenes used in earlier human studies. And so the selected dose range that we had was 2 and 4 grams.
Slide, Centella asiatica, Gotu-Kola, products. 3 images depict fresh leaves, commercial plant material, and dried Centella asiatica water extract, GA.
Now, this is what the CAW looks like. And so we're talking about giving people 4 grams of this.
Slide, product development. 2 photos appear, Kristen Wright, North Dakota, and Maya Caruso, BS. A photo appears of 3 glasses of liquid, 0 grams, 2 grams, and 4 grams of GAW. Text, delivery method, GAW drink mix powder. Excipients, dispersibility, palatability, safety, placebo matching, no TETs or CQAs.
And we decided that the best way to give it was as a drink, both because we hadn't done the plasma analysis then, but we wanted to be as close to the mouse studies as possible. And also 4 grams is kind of a lot to give in capsules. So the next thing was to try and develop a matching placebo to this drink mix and make the drink palatable and easy to disperse and so on. And so a lot of work went into formulating the placebo and the actual product, and that involved adding these excipients. And it was very important to make sure that the excipients that we added, which typically came from plant sources, didn't themselves contain the active compounds for like triterpenes and CQAs, because we didn't want them in our placebo.
Slide, is an investigational new drug designation needed? A flowchart lists data and specifications, leading to a box labeled yes, IND needed.
Then, of course, we had to see whether we needed an IND from the FDA to do our clinical studies. And there is a group at the FDA that you can ask when it comes to botanicals as to whether you need an IND or not. And they reviewed the kind of endpoints and the product and so on, and told us that we did in fact need an IND because, of course, as I mentioned at the beginning, our goal is to develop this as a botanical drug rather than as a dietary supplement. So for that reason, we had to get an IND.
Slide, FDA IND submission requirements. The title page of a document appears beside 9 section titles. A. Introductory statement and general investigative plan. B. Description of botanical product. C. Documentation of prior human experience. D. Chemistry, manufacturing and controls for the trial product, botanical raw material, drug substance, drug product. E. Non-clinical pharmacology/toxicity, preclinical studies. F. Clinical pharmacology, ADM studies and earlier clinical studies. G. Clinical considerations, anticipated risks, protection against risks, data safety and monitoring. H. Literature cited. I. Appendices, various including detailed trial protocol. Items C, D, and E are highlighted.
D. And the IND, there is guidance from the FDA for how to prepare and what you need to put in there. And the things I want to focus on are the documentation of prior human experience, describing how your product is made, and then also clinical pharmacology and toxicity.
Slide, a starburst image is labeled prior human experience with Centella asiatica, pointing to boxes such as marketed as tea, clinical trials of crude extracts, and publications on adverse effects.
So just skimming over these very quickly, we kind of were able to use data from previous clinical trials, as well as pointing out that people have been eating gotu kola for years, and then there's a lot of products out there that people are using with really very few adverse effects being reported in the FDA CARES database and so on.
Slide, product manufacture in CGMP compliant facilities and QC. A flowchart traces the path of the source Centella asiatica to stability studies. Logos beside each step list the partners involved: Wild Harvest, Ashland, OSU, and OSU.
We also had to go through this extensive process to make our product, which involved product manufacturing GMP facilities, and describe all this for our IND. And it also included stability studies on our product, which the FDA had required us to do.
Slide, organ toxicity not seen in mice treated with GAW. A photo of Randy Wilcher, MD, Ph.D., OSU, appears beside a flowchart, which illustrates a toxicity study on male and female mice. It outlines treatment dosages and durations, noting no elevation in serum ALT and AST levels. The chart concludes with histology results showing no abnormalities in major organs. A cautionary note about toxicity studies is included.
And then although we didn't do formal toxicity studies, we did actually analyze the organs from mice that had been treated with our Centella extracts in order to show that they didn't have any major abnormalities.
Slide, CAW has low potential for drug interactions. A publication title. Special section on natural products, experimental approaches to elucidate disposition mechanisms and predict pharmacokinetic drug interactions. Centella asiatica water extract shows low potential for cytochrome P450-mediated drug interactions.
So, and then we were able to, the FDA also wanted us to look into the potential for drug interactions, and we did this by contracting out some cytochrome P450 inhibition and induction studies to this company called BioIVT. And this data is also published, and luckily it appeared that CAW has low potential for drug interactions.
Slide, FDA IND application, approvals. A flowchart shows the path from the publications to clinical studies.
Having done all that, we put together our applications and got our IND, and were able to start our clinical trials.
Slide. Text contains scientific information about a bioanalytical method validation for a pharmacokinetic trial of a Centella asiatica extract in older adults. It lists details about oral absorption, including terpenes and metabolites. A separate section discusses antioxidants and pharmacokinetics in cognitively impaired older adults. Publication details appear beside a photo of Kristen Wright, North Dakota. Article, Pharmacokinetics and Pharmacodynamics of Key Components of a Standardized Centella asiatica Product in Cognitively Impaired Older Adults, a Phase 1, double-blind, randomized clinical trial.
And the first type of clinical trial were 2 pharmacokinetic studies, because we wanted to ensure that the active compounds from Centella would actually be absorbed and appear in the plasma of humans who were given our product.
Slide. Bioavailability of triterpenes from CAP in older adults. Line graphs compare the cognitively normal adults to impaired adults. Wright et al., Frontiers in Pharmacology, 2023. Wright et al., Antioxidants, 2022, N equals 4 participants.
And I'm happy to say that, in the 2 studies that we did, the cognitively normal humans and cognitively impaired humans, we were able to detect the triterpenes very robustly in their plasma.
Slide. A chart of values lists the pharmacokinetic parameters of components of Centella asiatica or their metabolites after administration of CAP containing Ca 2 grams or Ca 4 grams. Most analytes showed a significant difference in Cmax for the 2 grams and 4 grams doses. Wright et al., Antioxidants, 11, Article 215, 2022.
And we also found a lot of the CQAs and the CQA metabolites as well. So this kind of gave us confidence to carry on. And maybe Barbara's going to tell me that I've got 7 minutes left, I guess. Yeah. But we're getting there. Okay, so this was a very important study to do, and this is one that sometimes can be skipped, but I know that the NCCIH now is very keen that we do pharmacokinetic studies of any product that we propose to study in clinical studies, because we do need to show that the active compounds are absorbed.
The previous slide of line graphs appears.
And going back to our mouse chow versus drinking water study, where the way that we administered it seemed to have an effect on what was absorbed, this was doubly important to show that, in fact, the active compounds are absorbed into the plasma in humans.
Slide, current study, safety and target engagement by CAP. A flowchart outlines a clinical study involving older participants with MCI or mild AD. The study administers CAP or placebo for 6 weeks in a randomized, double-blind manner. It aims to assess biological signatures, safety, and tolerability. The chart includes brain imaging examples and lists various measures taken before and after treatment, including gene expression and cognitive assessments.
So currently we are running a clinical trial. So this is, again, not an efficacy trial yet, but we're currently running a clinical study in participants with mild cognitive impairment or mild Alzheimer's disease. They've been randomized to receive either the product containing the 4 grams of CAW or placebo, and they take that for 6 weeks. And in this study, we're not looking at efficacy. What we're really looking at is safety and tolerability. Of course, we want to make sure that when you give this product for a long time, longish period, that people are not experiencing adverse effects. But we're also trying to show that those mechanisms that we saw in the mice, which is basically a decrease in oxidative stress and improved mitochondrial activity and synaptic health, not synaptic health, but neuronal health, can we actually see this in humans when we give this extract for a similar period of time that we gave to the mice? And the way we're doing this is by measuring a marker of oxidative stress in the urine. And for brain mitochondrial activity and neuronal health, we're lucky to have an imaging core here at OHSU, where you can actually do magnetic resonance spectral imaging of the brain. And with the proton imaging, we're able to measure this compound called N-acetyl aspartic acid, which is a very specific marker for neuronal health in the brain. And we're hoping to see positive changes after 6 weeks of treatment with CAW. And we can also, using phosphorus MRSI, measure these high energy phosphates that relate to mitochondrial activity. And as I said, this study is still in process, so we don't know what we're going to see, but we're very excited to see what results this shows. And then of course, we're also using the opportunity to measure other things related to the possible activity of CAW, including some markers of neurodegeneration. And we're going to measure plasma levels, steady state plasma levels of centella triterpenes. And also we've included some cognitive measures, but we don't necessarily expect to see an effect, but it's worth looking while you're treating people. Now, if we get positive results from this will then directly go into an efficacy trial.
Slide, current status. Text, still actively recruiting. Text details the recruitment data, challenges for recruitment, and safety. A table lists the sources of randomized participants as of April 2025.
And what I want to say is, even having got to the point of doing this study, there's still many challenges because we're hoping to get 48 participants total. Currently, we have 33 randomized, so about 2/3 of the way along. But to get those 33, we had to start by identifying over 1,000 candidates and then put them through telephone screens and screening visits and so on. So this is, although it sounds like we've reached some kind of goal, the challenges continue. And particularly in this older age group, there's things like the fact that they have other health conditions and so on, and many of them have spare parts which are incompatible with the MRI and so on. But the good news is we haven't seen a lot of evidence of toxicity in the participants. And of course, currently they're still randomized. We don't know who's getting what, but fortunately, there have been no serious adverse effects.
Slide, progress toward Phase 3 clinical trials on the NCCE roadmap, de-risking steps. A diagram illustrates the building blocks of natural product clinical trials. It contains interconnected boxes highlighting various aspects such as pharmacokinetics, reproducible biological structure, recruitment strategies, and outcome measures. Red lines connect different elements, emphasizing their relationships within the overall clinical trial process.
So, take a deep breath. We feel that for Centella, we've actually ticked a lot of these boxes, and if we get positive results from these studies, we'll be able to go into at least single site and then multi-site efficacy trials. These trials will involve a lot more people and a lot more time, and therefore a lot more resources. So it's important that you've done all this groundwork so that you have the best possible chance of seeing an effect in your, or that, if it doesn't work, it's not because you didn't have the active compounds or you used the wrong type of product or it wasn't bioavailable. You've done all that groundwork to give yourself the best chance of seeing efficacy in this final trial or in the efficacy trial.
Slide, Centella asiatica, key new results at BENFRA. Text boxes list planned material, constituents, functional endpoints, and translatable biomarkers, with text detailing methods, improvements in health conditions, and brain activity.
Okay, and I just want to quickly mention that we've continued to do work on this at the BENFRA Center. Excitingly, we've shown that you can grow Centella asiatica in Central Oregon, and so you can grow it in the greenhouse, and we're hoping that as we move forward, we'll have our own source of Centella asiatica rather than having to rely on the commercial sources of variable quality. We've continued to explore the active compounds and also shown that Centella also has effects on things like mood, anxiety, sleep, and depression, and so on. These are some of the other studies that we've done. And we've also been looking at effects on vascular changes in the brain and other types of biomarkers that we could use in future studies.
Slide. Images list dietary supplement product containers for Centella asiatica.
So what does this mean for dietary supplements as a whole? As I said, a lot of our work has been focused on, developing a very specific product, the CAW, for cognitive function.
Slide, a flowchart illustrates the potential benefits of Centella asiatica, for cognitive function. It shows connections between known compounds, dosages, mechanisms of action, and possible applications for brain health and other conditions. Text, linking a botanical's biological effects to its chemistry is key.
But I hope that whatever we've done is going to be useful for the dietary supplement industry more widely, because we've shown that we have a good idea now of what active compounds are responsible for this, and therefore we are you can use these compounds as a handle for standardizing dietary supplements to at least kind of move towards a likely efficacious product. Also, because we know what the mechanisms of action are, this might be useful for other health conditions where these same mechanisms might be relevant. And what I want to emphasize is really understanding the link between the biological effects and chemistry is really key in this work. And so it's important to have that breadth of expertise for both biological and chemical effects.
Slides flash by quickly.
Now, I don't think I have a lot of time to talk about ashwagandha, but just to say that we've got this source, and we have a bunch of publications that relate to our development of analytical methods. The fact that we think that, like centella, it's not necessarily all to do with the most obvious compounds. Maybe there's compounds other than withanolides that are active. And also we've done some work in mice as well. We're a little less further along in this cycle, although in this pattern, but we do plan to get more data as we move forward with ashwagandha.
Slide. Our publications can be found at www.osu.edu/benfor-bdsrc. Screenshots depict the front page, and a list of publications.
If you're interested in our work, please visit the Benfra website, where both the current Benfra publications plus previous publications on our work are available. And to finish off, let me just thank all the very many collaborators and people who contributed to the work that I've described today, and everybody from the Benfra Center.
Slide. Dozens of small photos of collaborators is labeled the Benfra team. Names are listed under headers such as component leads, co-investigators, collaborators, and consultants.
We have a lot of people and we have a lot of fun.
Slide. An image displays a list of funding support sources for various research projects. It includes logos of organizations like NIH and the Alzheimer's Association. Grant numbers and researcher names are listed alongside specific funding initiatives and institutions, covering topics related to health, aging, and neurology research.
And of course, I have to thank the ODS, the NCCIH, and all our other funding sources that have provided the resources to allow us to do this work.
Slide. Thank you. Questions? 2 images of natural products are labeled Centella asiatica and Withania somnifera.
And finally, thank you for listening, and I'll be happy to take any questions. And that's my talk.
Amala, thank you so much. If you'd like to stop sharing.
Sure.
The 2 presenters' video feeds fill the screen.
One of the comments in the chat, a wonderful talk, an overview of the exacting science necessary to assess these herbal and botanical and other nutritional products, I would add to that comment.
Yeah.
Before we move on to the questions and answers, and I have some and there's some in the chat, I would like to introduce the brand new acting director of the Office of Dietary Supplements, Dr. Bremer. Drew, you should be able to unmute and turn on your camera now.
A third presenter's video feed appears.
I have. Thank you, Barbara. And now you're going to want to mute me and turn my camera off to having seen me. No, I want to thank Amala for a spectacular presentation. And I don't want to take time away from questions and answers, but what I would like to do is highlight 2 things. Highlight the import of Barbara Sorkin in having this series and having been a participant and watching for years now. It's just what a tour de force Barbara is, and I really am appreciative and grateful for her efforts in bringing this science to our knowledge. And Amala, you exemplify the strength and power of this seminar series, how we all learn from each other, and how as a collective whole it's more than just the sum of its parts. And I also want to thank everyone who's tuning in. I recognize that all of us have day jobs, and all of us are often stretched. So I do want to thank you for taking time to be a part of this and engage and encourage continued participation, because the answers and the discussion that we have at the end of these talks really do stimulate new ideas and really do help move the science forward. So with that, I wanted to thank you for the warm welcome. I am excited and humbled and honored to be a part of the OSD family, ODS family, and I look forward to many more talks. So Barbara, back to you.
Drew, thank you so much for the kind words. Thank you for
Yeah.
joining us, and welcome to ODS. I can tell that we're going to love having you with us. So with that, let's turn back to the science, and let me grab some of the questions.
So can I just say thank you so much to Dr. Bremer for joining us, for making time to attend this seminar. I'm really, truly grateful that he did that.
Wouldn't miss it. You bet.
Yes, to all the people who asked about sharing slides, and we hope we will be able to share the video. And then turning to, are you planning to? Will you need to do phase 2 clinical trials, or?
Yeah, I mean, definitely we would start small with a kind of single site study and do a phase 2 study before we go into multi-site. Absolutely, yeah. And I have to say that we have an Alzheimer's disease center here at OHSU who've been hugely supportive in this. So, and they've always been excited about this work. And so they're here to support us to do our first phase 2 single site study. So definitely that's it kind of depends what the results show as well, of course. But yes, that is in our plans.
I had 2 questions. One of them is in the clinical trial, you were giving the centella as a beverage, and yet in the mouse studies you found that it had very limited bioavailability in the drinking water. Any thoughts about why it's okay in the humans but not in the mice?
Oh, actually, it's the other way around, Barbara. We had better bioavailability with
in the water.
Mice that had it in the drinking water compared to giving it in the chow.
Right.
So
okay.
it kind of correlates with that. Yes.
Very conveniently.
Yeah.
Confused there.
That's okay. It is. There was a lot of data.
Well, while I'm talking, I don't want to miss other people's questions, but in the fly work, when you were looking at phototropism, do you think that you are actually restoring cognitive function to those flies, or were you feeding them throughout, so it could have been simply that you were protecting their cognition as they aged? Were they on centella the entire time?
So they started centella in their middle age, which is around 4 weeks of age. So they didn't have it from the time of eclosion, in other words, coming out of the larval stage or the pupal stage. They actually had it from 4 weeks to 6 weeks of age. And so, yes, I mean, it might have been preventing the decline. It's hard to know, of course, whether it's actually making them function better or it's actually stopping that process. But I would say that, compared to the mice, the flies that didn't receive the treatment, they did show better phototaxis. So and in parallel with the mice, it's not really a slowing down of the degeneration. It's an actual stimulation of cognitive function. It appears to be probably, again, through improved mitochondrial function or a combination of the two.
So do you see effects in younger animals in both cases as well?
The thing is, the effects are muted in younger flies and younger mice because they're already performing so well that it's hard to improve on perfect, basically, I guess.
You can't go beyond the level of mitochondrial activity.
you get a ceiling effect in a lot of these tests.
So we have a question about the methodologies that you use to verify identity and authenticity of plant materials. And could I make a comment here? You know that frequently for botanicals, a single active compound or group of active compounds, like the withanolides for ashwagandha, like the triterpenes for centella, are used as markers. And I think what you've shown us so beautifully today is that you're not going to get consistent activity if you monitor just the triterpenes for something like centella. So now the question,
thank you for that.
What methodologies do you use?
So we have been using a bunch of different methodologies. Most important, of course, we've been sending samples to the University of Mississippi, where they do genetic fingerprinting. So they compare the gene profile, the DNA profile of the materials we send to what's published, and then with other authentic samples. We also use simple methods like you can use microscopy. We don't use a lot of that, but we use a lot of thin layer chromatography where you get a profile of chemicals and you can compare that to authentic standards. So you can get authentic material, for example, from the American Herbal Pharmacopoeia, botanical reference materials. And I think there's some available through the NIH or other federal sources as well. You can buy botanical reference materials and compare to that on thin layer chromatography. And that's typically what we've done. We kind of start off with TLC and then also send samples to the University of Mississippi, who's one of the BENFRA collaborators for the genetic testing.
And then to make sure that you've got consistent product, you do the chemistry.
Oh yeah, that's right. The chemistry
Yeah.
for and then of course I should say LCMS. So that's not so much for identification, it's more for the quality, the quality of the thing. Although of course it does help with authentication as well, because if you found your, especially when you do the principal component analysis, if you found it was very skewed, that might be because it's got something that really shouldn't be there or the wrong chemical profile.
And then of course, very critically, especially with purchased products, checking for heavy metals and pesticides.
Yeah, that's right. Yeah.
Yeah. All right. What else have we got? We are running up on time. But there was a question. I don't know if you want to speak to the question on regulation of health claims on dietary supplement labels.
What is there a specific question? I mean,
the specific question is, could you clarify evidence is required to support substantiate claims made on dietary supplements is what the person.
My understanding is that you cannot make health claims on dietary supplement labels. And so the question of evidence doesn't come in. You can make structure function claims. So you could maybe say supports brain health, but you can't say improves, it can be used to treat Alzheimer's disease, for example, or prevents cognitive decline. You can't say that. But in fact, there are labels, websites and so on where you can get some guidance on what structure function claims you make. And it's my understanding that you don't need to substantiate that with evidence. Anything that you use as a structure function claim doesn't necessarily have to have any kind of substantiation. If anyone's here from the FDA or regulatory bodies or herbal associations and can add to that, I'd be happy to hear about that.
And Dr. Gupta, I'm sorry, I'm having trouble making you a presenter so that you can unmute. Could you put your question in the chat? So sorry.
I just gave them permission to unmute.
There you go. Okay, Dr. Gupta, please go ahead with your question.
Well, my question is that I have been a part of the USDA NIFA, and I am also working on the nutrigenomics that Dr. Soumya was mentioning about. I would like to know if there is any partnership available for anybody, and I can send out the email, to Dr. Soumya as well. And I think that there was an email there to send. And I would be really interested because I'm a similar kind of work I've been doing for quite many years now. So I'm looking for somebody who can guide me in that area. Although I've been a part of CBG, USDA, and NIFA, and got a couple of grants from there, but I'm trying to get this thing clinical trial, just like the way Dr. Soumya is mentioning. So I would be very interested to partner with someone who is, in that area. Thank you.
That's fantastic. Thank you for stating that. I've just put my email in the chat. So if anyone would like to email me, please do that.
And both Dr. Rai and Dr. Casper have pointed out that even for structure function claims, manufacturers do need to have substantiation data in their files.
Okay, thank you.
So, okay, and we are past time. So thank you all for the great questions. Thank you all for joining us. And thank you so much, Amala, for stitching together this amazing team that did all of this amazing work.
Yeah.
And for being so generous with your time and your knowledge today.
Oh, thank you so much for the opportunity. And thank you everyone who attended the talk from me.
Thank you.
Thanks.
See you all next month, I hope.
Yeah. Thanks.